forgotten antibiotic can help in the fight against drug-resistant pathogens. Scientists have been waging war against drug-resistant bacteria for years. Perhaps a new weapon will come into their hands soon. It is a forgotten antibiotic that was isolated from soil bacteria back in the 1940s. It turns out to be extremely effective in the fight against superbugs. Half of today's bacteria-killing drugs are variations of compounds discovered decades ago, during the golden age of antibiotics. Also then, a substance called streptothricin was isolated. It is extremely effective against gram-negative bacteria. Previously, the drug was not introduced to the market because it showed high toxicity. However, new research on one variant of the compound gives hope for its wide application in medicine. Unlike gram-positive bacteria, gram-negative microbes do not have a solid cell wall, which is affected by many known antibiotics. Finding a drug that fights these bacteria is one of the greatest challenges facing the pharmaceutical industry. This is evidenced by the list of the most dangerous pathogens published in 2017 by the World Health Organization, WHO. Gram-negative bacteria make up the majority of the list. Description and results of new research on streptothricin appeared in the journal PLOS Biology. After streptothricin was isolated, scientists noticed that it was very effective in fighting gram-negative bacteria. However, the drug was not put on the market because it was considered too toxic for human kidneys. For this reason, the substance remained hidden in the scientific literature. Now James Kirby of Harvard University and his team have returned to research on the potential of streptothricin. With the emergence of drug-resistant pathogens that are ineffective against known antibiotics, it's time to revisit and explore the potential of what we may have previously overlooked, explains Kirby. The substance analyzed by the researchers is called norsotricin. It mainly contains two compounds, streptothricin D and streptothricin F. It is produced by actinomycetes, gram-positive bacteria living in the soil. Scientists have been able to determine that streptothricin D actually has a strong toxic effect on kidney cells. However, it is different in the case of streptothricin F. This compound effectively kills drug-resistant gram-negative bacteria and does not have such a negative effect on human cells. The effectiveness of the drug was confirmed in studies on mice. Streptothricin F managed to kill the drug-resistant strain of bacteria with minimal or no toxicity. Bacteria living in the soil have been trying to maintain their territory for millions of years. In the course of evolution, they figured out how to produce antibiotics that can penetrate the armor of gram-negative bacteria. Streptothricins are one of the results of this ongoing arms race, says Kirby. According to the scientist, the analyzed streptothricins D and F specifically fight the defense mechanisms of gram-negative bacteria. Streptothricin F attaches to the bacterial ribosome in a different way than in the case of known and used bactericides for years. Although researchers have not yet learned the details of this mechanism. However, a different way of binding streptothricin F to the ribosome gives hope that it may work once other possible therapies have been exhausted. If scientists manage to understand the mechanisms of action of streptothricin, it could lead to the development of a completely new class of drugs that will effectively fight superbugs. Currently, researchers are continuing to analyze streptothricin. They hope that the world of science will become interested in this substance discovered 80 years ago. New findings on the largest mass extinction. The toxic soup killed most of the animals. At the end of the Permian, about 252 million years ago, the worst mass extinctions of animals and plants in history occurred. Scientists have linked him to massive volcanic eruptions and the resulting greenhouse gas emissions, which have led to a significant increase in the Earth's temperature. Life on our planet has almost completely died out. Scientists have determined the details of the greatest catastrophe in history. Moreover, they point to many parallels between the Permian extinction and today. An international team of scientists, including Professor Tracy Frank and Professor Chris Fielding, identified a new cause of the Permian extinction. It's about toxic blooms of microorganisms that were deadly to animals, we read in an article published in Nature Communications. In a healthy ecosystem, microscopic algae and cyanobacteria provide oxygen to aquatic animals through the process of photosynthesis. However, when their numbers get out of hand, these organisms can reduce oxygen in the water and even release toxins into the environment. By examining sediments and chemical records from rocks originating in Australia, the researchers found that out-of-control blooms occurred shortly after the first volcanic eruptions that contributed to the late Permian mass extinction. When the algae-eating animals died out, there was no one left to keep their blooms in check. Freshwater rivers and lakes became infested with toxic bacteria, delaying the return and evolution of organisms by millions of years. The Permian extinction is the largest known species extinction in history. In the last few million years of this period, about 90 to 95% died out. Marine organisms, about 60%. Families of reptiles and amphibians by about 30%. Rows of insects. The Permian extinction also affected ferns, horsetails and club moss. Professor Frank and Professor Fielding are engaged in the study of sediments. Now they have analyzed layers from the time of the Permian extinction and have discovered new evidence of toxic soup in rivers and lakes at that time. By examining the sediments, we try to discover the conditions in which animals and algae lived at that time, explains Professor Frank. For example, we can determine whether the sediments come from a lake or a river, the salinity level and the temperature of the waters, he adds. 3 elements are needed for the toxic soup to appear in the waters: rapid emission of greenhouse gases, high temperature and abundance of nutrients. Volcanic eruptions caused gas emissions and temperature rises, and when the trees were swept away, the soil leaked into rivers and lakes, providing all the nutrients the microbes needed. The researchers compared fossil records from the times of various warming-related mass extinctions. As it turned out, similar deadly algae blooms always occurred then and were the cause of extinction of organisms living in fresh water. Currently, microbial blooms are becoming more frequent. According to the researchers, thanks to the geological sciences, we can better understand what is happening today. We are seeing more and more toxic algae blooms in lakes and shallow seashores, which is related to the increase in temperature and the increasing amount of nutrients in the waters, says Professor Frank. There are many similarities between the Permian extinction and today. The rate of increase in the amount of carbon dioxide in the atmosphere is similar, although then volcanoes were responsible for it, and today it is man, adds the researcher. Thanks to our research, we can better understand how much the climate has changed in the past, what were the climate extremes at that time, how fast the changes were taking place, and what were their causes. This gives us a good background to understand what is happening today, emphasizes Professor Frank. According to this year's report by the Intergovernmental Panel on Climate Change, IPCC. The human impact on climate change is unequivocal. By their actions, people create conditions that are conducive to the spread of thermophilic algae and cyanobacteria. Combined with the influx of nutrients from water pollution from agriculture and deforestation, this leads to a surge in toxic blooms. As a result, fish die, human and animal health is at risk, and billions of dollars in damage are caused. The end of the Permian is one of the best periods to look for analogies with what is happening now, argues Professor Fielding. Another similarity is that the increase in temperature 250 million years ago coincided with huge forest fires that destroyed entire ecosystems. We are seeing similar phenomena now in places like California. One wonders what the long-term consequences of such events are, as they become more and more common, the scientist adds. According to researchers, the balance of many ecosystems on our planet is currently disturbed. However, humanity has the ability to prevent global warming. For example, by reducing greenhouse gas emissions. The scary thing is that we're used to thinking in terms of years, maybe decades. However, the mass extinction at the end of the Permian lasted 4 million years. This should be sobering for people, emphasizes Professor Fielding. Scientists have fully read the human genome. A group of scientists in the Telomere to Telomere (T2T) consortium has published the first complete, gap-free sequence of the human genome. This knowledge is crucial for understanding the basis of various diseases, human genetic variability and evolution. In 6 papers published in the journal Science, scientists described the painstaking work of deciphering the human genome. This work was carried out as part of the Telomere to Telomere (T2T), the name taken from the ends of the chromosomes consortium, which included researchers from, among others, DRIVE. From the National Human Genome Research Institute, NHGRI, which is part of the National Institutes of Health, scientists from the University of California, Santa Cruz and the University of Washington in Seattle. The research was led by Adam Philippi of the National Institutes of Health. The first publications on reading the human genome appeared over 20 years ago. It was a huge achievement and the researchers of the Human Genome Project were behind it. The deciphered genome covered most of the protein coding regions, but about 8% of the genome was missing to be sequenced. It took 2 more decades to fully understand the human genome. The difficulties were mainly due to the fact that the missing 8% are repetitive pieces of DNA that are difficult to match up with the rest. The technology at the time couldn't handle it. However, there have been significant advances in sequencing methods over the last 20 years. Thousands of researchers from various institutions spread around the world have developed better laboratory tools, computational methods and approaches to decipher a complex sequence. This was helped by a special cell line that has 2 identical copies of each chromosome, unlike most human cells, which have 2 slightly different copies. Until recently, sequencing consisted of breaking the entire genome into smaller pieces and reading those smaller fragments. However, using this method leaves some gaps in the assembled genome sequences. Because these sequences are repetitive and contain many duplicate regions, trying to put them in the right places can be compared to putting together a puzzle where all the pieces are the same shape and have no picture on the front. Sequencing machines and genome assembly software could not penetrate areas where the DNA sequence contained highly repetitive stretches. Repetitions could easily be missed or incorrectly connected, which accumulated errors. As sequencing technology advanced and costs fell, scientists reduced the number of gaps or incorrectly assembled sequences, culminating in the publication in 2017 of a reference genome called GRCH38. They became the basis of work and reference for many, but still contained gaps. However, over the past decade, new sequencing technologies have emerged that have allowed the analysis of much larger stretches of DNA, allowing the discovery of previously overlooked fragments. A DNA sequencing method called Oxford Nanopore can read up to 1 million letters of DNA in a single read, but the accuracy of this technique is not the greatest. But another method, PacBio Hi-Fi, can read about 20k letters with near-perfect accuracy. Researchers in the T2T consortium used both DNA sequencing methods to generate a complete human genome sequence. The new reference genome, called T2TCHM13, will be made publicly available. Generating a truly complete human genome sequence is an amazing scientific achievement, providing the first comprehensive picture of our DNA, said Dr. Eric Green, director of the NHGRI. This fundamental information will reinforce the many ongoing efforts to understand all the functional nuances of the human genome, he added. Since we had the first draft of the human genome sequence, determining the exact sequence of complex genome regions has been a challenge, said Dr. Evan Eichler of the University of Washington School of Medicine and co-leader of the T2T consortium. I am thrilled that we were able to complete this task. The complete genome will revolutionize the way we think about human genome variability, disease and evolution, he added. The researchers noted that most of the newly added DNA sequences are located near repeating telomeres, the long ends of each chromosome, and centromeres, the dense middle sections of each chromosome. Previously unreadable genome sequences that have now been put in the right place and sequence are critical to understanding the full spectrum of variation in the human genome and understanding the genetic contribution to certain diseases. The complete version consists of the 3.055 billion base pairs that make up the human genome. Researchers also identified 1,900 previously unknown genes. They also used the now complete genome sequence to discover more than 2 million additional variants in the human genome. These studies provide more accurate information about genomic variants in 622 medically significant genes. In the future, when someone sequences their genome, we will be able to identify all the variants in their DNA and use that information to better guide their healthcare, said Dr. Philippi. The true completion of the human genome sequence is like putting on a new pair of glasses. Now that we can see everything clearly, we are one step closer to understanding what it all means. Parts of the genome with many repeating stretches include those where most human genetic variation occurs. The variability in these regions may also provide key clues about how our ancestors underwent rapid evolutionary changes that led to who we are today. While sequencing each centromere, they learned that the duplicated regions vary greatly in size, which was quite unexpected. We believe that when studying these new parts of the genome, there will be genetic variation contributing to many different traits and disease risk, said Rajiv McCoy of Johns Hopkins University, a member of the T2T consortium. In general, we don't know what we don't know yet, he added.